Okay, if I could have your attention, I'd like to get started with our next talk, continuing our afternoon session. So our next speaker is Tom Kepler. He's a professor of microbiology at Boston University School of Medicine. He's also director for the Center for Computational Immunology located at Boston University with ties to Duke, which is again one of the centers for modeling immunity for biodefense. He got his BA at the University of Massachusetts in Boston and a PhD from Brandeis University. He continued some postdoctoral work at Brandeis and then also postdoctoral studies at the Santa Fe Institute. He then was at the Department of Statistics at North Carolina State University for a number of years, and then the Department of Biostatistics at Duke prior to moving relatively recently to Boston University. And in the Kepler laboratory, they developed computational tools, applying them in the context of systems-level experimentation, again, that common theme of relating experiment and computation, and trying to address questions in immunology, vaccine development, and in conjunction with partners at Duke and Harvard universities, they developed a new approach to vaccination that applies some computational methods, and it has to do with driving affinity maturation and so on. And I suspect that's some of what we'll hear about today as he talks about analysis of antibody clonal dynamics. Tom, it's a pleasure to have you here.
Thanks, Dave. It is great to be back in Blacksburg. My son got his undergraduate degree here, and he's a big fan of Virginia Tech, and I think that he will be for the rest of his days. He really enjoyed himself here. Yeah, I will be talking about affinity maturation and the computational tools that we've developed to help understand affinity maturation, with the ultimate goal of helping us to induce affinity maturation in the context of vaccines. And the structure of my talk is going to really be a series of vignettes, more than a single coherent presentation from beginning to end. And that gives me the advantage that if I run out of time, I can just chop off the last vignette. And that does happen from time to time. So I'm going to go over the background that you're going to need to understand to appreciate what we've been doing. And in fact, I'm going to go back into the history a little bit more than is usual, simply because I really enjoy it, and I hope to convey that sense to you. Then I'll describe the statistical methods that my group has developed to help us understand affinity maturation. And then I'll go through three specific examples of where we use these methods. One is in looking at the response to an experimental influenza infection, looking at the B-cell response. One is looking at the evolution of a so-called broadly neutralizing antibody against HIV-1. And then finally, still looking at HIV-1, we'll begin to look at the role of insertions and deletions in addition to simple point mutations. And we'll start by going back into the history of it. So one of my dreams or my long-term goals is to put together a model of a particular system, a biological system, in which the model is able to grow organically as new knowledge is acquired. And I think that the best way to begin thinking about how to do that is to understand historically how knowledge has grown in a particular field, to understand what changes were wrought by individual discoveries and individual experimental results. And imagine if you had been trying to put together a dynamical systems model, your whatever favorite kind of model you have at the time. What would the model have to have looked like in order for it to have grown organically from that point? And so I'm going to just go through just a couple of pieces of background that you would have had to see anyway, only I'm going to try to put it into the context in which it was first discovered at the time. So please bear with me for this minor indulgence. Went the wrong way because I'm looking at the wrong machine, actually. So I was going over the overview in my mind. You weren't seeing this. I was seeing it there, so I apologize. I'm going to give you another 5 seconds to look at it since you haven't seen it before. 2, 1, and so now on to the background and history. This is the front page of a German medical journal from 1890, and in this journal, von Behring and Kitasato report some interesting results that they acquired with rabbits immunized with with tetanus toxoid. And so it's really simple experiment, but it's exceedingly beautiful. They immunized a rabbit with tetanus toxoid. So tetanus toxoid is just a slightly less lethal form of tetanus toxin. And the rabbit was thereafter protected from tetanus infection by the organism and by intoxication by tetanus toxin. That was already well understood. But what they did next was much more interesting. They took serum from the rabbit, the immunized rabbit, injected it into an unvaccinated rabbit, and lo and behold, the other rabbit was also protected from infection and intoxication. So what they had shown was that there was something in the serum of the first rabbit that conveyed protection to the second rabbit. And so what they had done was isolated a protective factor that we would then go on to discover were antibodies. At this time, of course, that wasn't known, but subsequent experiments showed that you could protect rabbits against virtually anything through the same procedure. And also, if you immunized against pertussis, then it wouldn't protect against tetanus and vice versa. And in the subsequent decades, it was found that you could make antibodies to just about anything. And so the next question arose is, how are you able to encode such specificity? They thought there must be thousands or millions of different specificities. How is that possible? And that was not discovered until almost 100 years later, 1978, when Susumu Tonegawa studied the immunoglobulin loci. Well, they weren't loci at the time. He had transcripts and looked to see where the transcript hybridized to in 2 different kinds of cells, an embryo cell and a plasma cell, a cell which had already by that time been known to make antibody. And they found that the hybridization patterns were quite different, and from that they deduced, Tonegawa deduced, that what was going on was that there were 2 segments in the gene, and that they were separated in the genome, but then the cells that would go ahead and make antibody would join these 2 gene segments together to make a complete antibody. And of course, that's now what I think is truly the hallmark of adaptive immunity, is the ability to recombine receptors. And of course, we've learned a lot more about that. I keep going back to that one. And of course, for heavy chains, which is what I'll be talking about for the most part, for the remainder of the talk, not only are there V segments and J segments, but there are D segments as well. And so just to refresh, to make an antibody heavy chain, you pick a V segment, a D segment, and a J segment, stochastically, not completely at random, there are biases, and you make a complete heavy chain VDJ rearrangement. Now, in addition, and this is something that we'll just have to bear in mind for what is to come, there is further stochasticity in the exact location of the place where each of the gene segments is cut. And just to make it all the more interesting, there are non-templated nucleotides that are polymerized into the junction. Okay, so altogether we have combinatorial diversity in the selection of the gene segments. We have diversity in the location of the individual recombination points, and we have diversity in the insertion of non-templated or N nucleotides. We'll come back to that momentarily. So the cells that bear, well, that make and bear these molecules, antibody molecules, are called B cells. And what's interesting is that in 1965 and 1966, Max Cooper was studying the B cells and T cells in chickens. And what Max discovered was that if you cut out the thymus of a chicken, you would lose a lot of the functioning of the immune system, but you didn't lose antibodies. And if you cut out the bursa of fabricus of the chicken, you would lose antibodies, but not a lot of the other stuff. And so, in so doing, he really laid out the grounds for separating the two lineages of B cells and T cells. And of course, now we know that they have to cooperate with each other, so that the antibody is worn as a receptor on the B cell. And of course, remember, these receptors are created stochastically, and so they have a great variety of different binding sites. Once they do bind their ligands on, for example, a bacterial surface or on the surface of an infected cell or whatever it is, they process the antigen presented to T cells, and then there's a mutual signaling that activates the B cell, and the B cell then differentiates to become a plasma cell, perhaps, or a memory cell. We'll talk about that in a minute. And then the B cell secretes antibody of the same type. So it's a very clever system. And then the antibody goes on to serve several different important functions. Now, even before we understood how rearrangement occurred, it was noticed in just about the same time that Max Cooper was figuring out the difference between B cells and T cells, that if you immunized a rabbit, rabbits played a big role in the early days of immunology, apparently. If you immunized a rabbit with an artificial hapten, and then measured the affinity of the serum antibody for that hapten complex over the next few weeks, something really extraordinary happened. And that is that, and I'm just pointing out one specific row, but all the rows tell a somewhat similar story, the affinity of the serum for the hapten goes from 1 at 2 weeks, 6-fold higher to 5.9 at 5 weeks, and then up to 250 in 8 weeks. So this affinity is growing really substantially. And it's really quite an extraordinary story, and we had no idea what was going on at that point. But we knew that the affinity was changing, and so Hermann Eisen called it affinity maturation. 20 years after that, we started to understand how it worked. And this was work done in Martin Weigert's laboratory. He was again looking at light chains, and at this point, sequencing was becoming more widely used. And so he sequenced a handful of kappa genes, and what he discovered in trying to make sense of what he saw is he noticed that the genes differed from each other by just a few point mutations, and in fact, you could build a phylogenetic tree. Oops. You could build a phylogenetic tree from the sequences that he recovered. And so he said, this is really strong evidence, as far as I'm concerned, for the introduction of mutations during the course of the immune response following immunization. In other words, we immunize, and then the genes that encode the antibody molecules go through a series of steps in which mutations are introduced, and he had no evidence for it at the time, but he said it must be that these mutations that introduce higher affinity antibodies confer an adaptive advantage to the B cells that bear them. And so that's how affinity maturation works. Point mutations and then selection. And that turned out to be quite right. And now we know very clearly that affinity maturation through these processes is really the key to getting a good vaccine response. And so here's an example from a laboratory of colleagues with whom I was collaborating at the time at Duke. What we did was immunize mice with a protein from Bacillus anthracis called protective antigen, or PA. And we measured the affinity over time, over a 3-month period, so this is whole serum, either where there was no immunization, where there's immunization with the protein alone, or immunization with the protein plus a potent adjuvant. And we were finding that the affinity rose more rapidly and ultimately reached higher levels if you used an adjuvant. And so what we're interested really is understanding how we can drive this higher affinity response through means like adjuvants. Why do we care about affinity? It turns out that high affinity typically means better function. And here again is data from the Sempowski-Alam group at Duke. What they did was from the same system that we were looking at, they took serum, they measured the average affinity in the serum, and they looked at the neutralization capacity, the ability to neutralize lethal toxin in macrophages, and they found that the higher the affinity, the better the neutralization. So that's basically the story. High affinity, good. High affinity comes from somatic mutation selection. How do we get a hold of those processes to drive the kinds of responses that we want? That's what this is going to be about. And so I've set up the background. Now I'm going to do some of the math, some of the statistics. And then this workshop is about modeling. And you have to make sure that you don't blink to miss the model in the talk. Because my model is a statistical model. Okay, it's genuinely a model for reasons that I'll describe. But it's not long sets of differential equations. I do those too. But what I'm going to talk about today is a more subtle kind of modeling. It's a statistical model. And so what we need to do is to account for 2 different sources of variability in immunoglobulin genes. The first source is in the initial rearrangement process. The stochastic process gives rise to a given initial gene. And it's that gene that the naive B cells have when they first pop out into the periphery. And that process is called rearrangement. So we have to have a model for rearrangement, and then we have a somewhat more complicated model for evolution by somatic mutation. And when I put these together, I'll have a model for the process of affinity maturation. Although some key features will be left out for another grant proposal. So let's talk about the arrangement process itself. Remember, there are a very small number of things that you have to tell me, and I can tell you what the resulting gene looks like. You have to tell me what the V gene segment is, the D and the J gene segments. You have to tell me the places where the segments are cut, and then you have to tell me what nucleotides you've put into the junctions. And so this is a gene, a heavy chain, human heavy chain gene that we've got in one of our anthrax vaccine studies. And we've got it broken down into the maximum likelihood configuration. So in other words, what we do is we estimate parameters, just like any other statistical estimation procedure. And in this particular case, it says that the result was that the most likely choice for V gene segment is a V gene segment called VH1-3*01. And it looks as if the V gene is cut one position from the end. And we say that because you notice that all of these positions match. The dot indicates a match between the observed sequence and the germline VH gene segment, but it doesn't match here. So we figure this is probably where it's cut. And it's possible, of course, that it actually was originally encoded by this nucleotide, and so this was an A, but it mutated. It's possible. But it's less likely. And similarly, the best reconstruction, the best estimate says that all of these nucleotides come from N nucleotide addition. And then similarly, the D gene segment starts here. There's a mutation in the middle, another mutation here, a short N region, and then J starts here. So that's what you have to do. For every one of these things, you have to estimate the best rearrangement. Trouble is that the second best rearrangements are often not that bad either. So here's another example. So the next two best rearrangements have the same 5' end, but down here, this one says, well, the place where D is cut is actually a little bit deeper. And when you do that, then this is no longer a mutation because it was originally encoded by D. And you pay for it by saying, I have to have more N nucleotides, and so forth. And so what we're going to want to do is to account not only for the top scoring rearrangement, but for all of them. And to do that, we're going to use Bayesian statistics. And if you aren't familiar with Bayesian statistics, the basic idea is don't just try to find the best parameter set, but average over all the parameter sets. And in general, if you don't know something, average over everything, weighting it by the probability that it's the correct one. That's Bayesian is in a nutshell, and that's the way we're going to be proceeding. So here's an example of a case where it seems absolutely essential to use Bayesian procedures because the best possible rearrangement, the best possible story for how this antibody gene came about is not much better at all than the second best, and so forth. So this is the same kind of diagram showing the V, D, and the J aligned underneath the observed sequence. This sequence is called VRCO1, and you see it's just really hard to decide where the recombination points are. And therefore it's going to be really hard to decide exactly what this gene looked like before it acquired these mutations. Why do I care? Why do I make a big deal out of VRCO1? Well, VRCO1 is currently the most potent and most broadly neutralizing antibody against HIV-1 that we know of. It is 32% mutated at the nucleotide level. This is an extraordinary level of mutation. And what this tells us, well, in addition to all of the other broadly neutralizing antibodies that we've found, they all have a high level of mutation. What this tells us is that affinity maturation plays a key role in the development of broadly neutralizing antibodies against HIV. And this is why we have to study affinity maturation all the more intently if we're going to make a good vaccine against HIV. So it's an important antibody. It's clearly driven by affinity maturation, but unfortunately, we can't really study the process in this antibody because all of the mutations that it has acquired have essentially completely obscured its history. So this one, in spite of its importance, is going to be very difficult to study. And it tells us that we have to do much more careful statistical modeling than perhaps people might have thought. So the second part of the model, we've done rearrangement. We have a model for rearrangement. It seems just completely trivial. You just pick a bunch of parameter values, and that's your rearrangement. The next part is a little bit more interesting. The next part is genuinely dynamic. It's a Markov model for the introduction of point mutations. And so we're going to take each column in the alignment, every nucleotide position, as independent of all the rest, which is what one typically does in phylogenetic statistics. And the key part of our model is going to be a function that gives the probability that a nucleotide in state A for ancestral, after a passage of t units of time, will be observed to be in state n. Okay, so A goes to n after a period of time t, and it's a probability, and it's very straightforward. There are a number of different models that you can use, depending on the level of complexity that you want, but this is all there is. Now you put all of these together, string them side by side along the entire length of the molecule, and that's your model. So putting it all together, we have a model for somatic hypermutation and for rearrangement. And now we've got an interesting extension of the methods that people have used for many decades, well, for 20, 30 years, building maximum likelihood phylogenetic trees. So the basic idea is that I've got 3 sequences that I believe belong to the same clone. That is, the same B cell was stimulated and divided and mutated along the way and made 3 descendants that differ from each other by mutations. And we build a phylogenetic tree showing how they're related. And then what we want to do is to guess what the original form of the ancestor was. That's what the first part of our procedure is going to be. What was the original ancestor? What did it look like here? Well, if I'm just doing maximum likelihood and I've got a bunch of proteins that I'm trying to decide their ancestor for, we have no constraints on what the ancestor might be. And so for a gene of length 300, it's an astronomical number of possibilities. But if it's an immunoglobulin, the interesting thing is that the same setup gives you only about 10 to the 12th different possibilities, because there are only that many different immunoglobulin unmutated rearrangements that can occur, roughly speaking, with the appropriate probabilistic weighting and so forth. So what we thought of as an extraordinary, and people use words like nearly limitless or nearly infinite diversity of immunoglobulins, turns out to be an extremely small subset of all possible amino acid sequences, which is a really, really great thing for us. That means that we can actually run through all of the possible ancestors here and do a complete probabilistic Bayesian inference and decide which ones make the best ancestors. And that's exactly what we do. And you notice that we can do that for whole sets of clonally related sequences by these techniques. And that's going to be the key. So here's an example. I won't dwell on this example. This is a clone that was published a few years ago. And so we took these sequences and reconstructed the clonal ancestry of it. And this is impossible for you to see, but I know that there are just a couple of things that I want you to see. This is the region called CDR3, complementarity determining region 3, which forms a crucial part of the antigen binding domain. And what we're looking at here is the estimated error at that position. And so in CDR3, we have a few positions where the error is around 15 to 20%. And these positions tend to be places where we are undecided about whether a position was originally encoded by a D gene or by an N nucleotide. So we've got this kind of slop. Here, this error is 50%, and this is due to allelic differences. So there are 2 VH genes that encode the observed clonal sequences equally well, but they have allelic differences that we can't resolve on this data alone. And so these remain. Point here is really just to point out that there really are residual uncertainties that need to be resolved, and we'll come back to that in a little bit as well. So here's our VRC01. This time we're looking at it, again, this CDR3, and there are 4 possible nucleotide states, C, T, G, and A. And what I'm showing here is at each position, the posterior probability that the nucleotide state is given by each of these 4. And so here, this is the invariant cysteine, this is the codon for cysteine, and this must be here. And so we have TGT, and they're bright white. We have very high confidence that these are correct. In fact, we know that they have to be correct, or else it wouldn't have made an antibody. But then here in the middle of CDR3, we have a great deal more uncertainty. And so if we're trying to understand the history of this antibody, we're going to be in deep trouble, because we can't reconstruct it at all adequately. But there are those that we can reconstruct, because we have better information, and the antibodies themselves have lost far less information over the course of their evolution. And this is one study of a clone called 2569 that was obtained by Tony Moody and his collaborators from Duke in a study of experimental influenza infection. So this study was conducted in the UK. I don't know whether it could be done in the United States or not. They had volunteers who agreed to inhale live influenza. And they got sick. And 7 days after they got sick, they gave blood samples. And antibodies were recovered from these blood samples. Genetics was done, and then they gave these antibody sequences to me for my processing. And so out of a few hundred of these sequences, we found several sequences that appeared to be clonally related, that is, have descended from the same initial B cell. And there was one that allowed us to infer the history particularly well, that is, with high confidence. And so we studied this one. And this technology is no longer quite as cutting edge as it was 2 years ago when I started talking about this work. But it's still, I think it's impressive. And if you think about going back to Kiyosaro and von Behring, how far we've come in technology. Here we're taking individual B cells, putting them one B cell at a time into individual wells, and then cloning out the heavy chain and light chain from single cells simultaneously. And so what we end up with is a light chain, heavy chain pair for each of these individual B cells. Then we can transfect these B cells into cells that make the antibody, and we can study the properties of those antibodies as a pure protein system. It's a monoclonal antibody at this point. But the really cool thing, as far as I'm concerned, is this is all based on sequence. They get the sequence, then they make the vectors, and then they make the antibodies, which means that I can put anything I want to into the vector. And in particular, I can make my phylogram the way that I described, and starting with the 5 observed sequences, I can also infer what the ancestor looked like and what the intermediate antibodies looked like by their positions on the tree. I can run my evolution backwards or between these two, and I get very good inferences for these. And so I can make all of these antibodies. So Larry Liao at Duke synthesized the antibodies that our group inferred, and Munir Alam computed or measured the affinity. And what we found was that if you plot the dissociation constant, which is the inverse of the affinity, against the evolutionary distance measured in expected substitutions per position, you have a very nice linear regression in the log of the dissociation constant. In other words, over the course of this evolution, the affinity is rising exponentially and doing so at a very consistent rate. And I thought this was a really remarkable finding that it was so continuous in its pace. The other thing that's really interesting is that what we ended up with is 5 different sequences, all of which had pretty good affinity. And if you look back at the sequences that we've got here, they're very different from each other. In fact, they are, oh, I guess I don't have that here. Sorry about that. They differ from each other by several amino acids each. And I'm sorry that I don't remember the exact numbers. So there are 5 different solutions to this problem. The other thing that we can do is to look and see where the mutations accumulate. And this is going to be the most taxing part of the presentation. So if your attention is going to wax and wane, I think it should wax right here. Okay, pay attention just for 2 slides. Because if you haven't worried about somatic mutation before, this will be sort of different. So let's focus our attention here first. This is a histogram along the length of the heavy chain and along the length of the light chain. And this is just a histogram showing where the mutations fall. And this is showing where the mutations fall by evolutionary distance. So each of these observations occurs at a given distance, and we put the mutations in where they appear. And so we've got these interesting patterns. And we ask, well, do they correspond to anything? Well, here's something that's important about somatic mutation. Somatic mutation is introduced non-uniformly along the length of the immunoglobulin gene. There are certain places in the gene that get mutated much more readily than other places. And it has to do with the nucleotide sequence that binds the enzyme AID. AID is the enzyme that is responsible for making the initial lesions in the DNA that then cause mutations to occur. And so where AID is more likely to bind, that's where you're more likely to have mutations. And so what we're showing here is the plot of where AID is more likely to bind compared to the average. So the average is 0, and in places where the lines go upward, this is where AID is more likely to bind. Where they go downward, it's where it's unlikely to bind, where there are cold spots. So this is sort of temperature, if you like. More likely to mutate, less likely to mutate. And these 3 regions here show where the complementarity determining regions are. These are the regions that encode the parts of the antibody molecule that actually bind the antigen. And so you see that the first 2 CDRs seem to be, a good bit hotter than their surroundings. CDR3 not so much, and interestingly, there's a region here in what's called framework 3 that seems to be fairly mutable in spite of the fact that it doesn't bind antigen. And now let's look at the observed distribution of mutations, and you see that it pretty much follows these the natural tendencies of the mutator. And what we're looking at here is both expressed and synonymous mutations. And this histogram is for expressed mutations only. And in particular, you look here at framework 3, these mutations are accumulating here, even though they're not likely to be playing a role in antigen binding. So it's a sort of a strange thing. So then we can ask the question, why do these mutations have these patterns? Why do they fall in CDRs and not in the frameworks? And this is a question that I and others have addressed in the past. And the answer is basically that these sequences have evolved so as to put mutable nucleotides in places where they might do some good. And where they're likely to do harm, they've made the positions less mutable. And this is by manipulating codon bias, codon usage. So if you need a particular amino acid like serine here, then what you're going to do is you're going to find the least mutable serine codon you can to put it there. If you need serine here, you'll use the most mutable codon. And so that's what's happened over evolution. But now that we've got a specific clone and we can measure the affinities and so forth, we can ask an even more pointed question. If I'm starting at a particular position in sequence space, okay, here's my antibody gene in sequence space, and what the mutations are going to do is to move me around in this sequence space. And I move, of course, they're discrete jumps from one nucleotide to another at a particular position. And so if I have 300 nucleotide long sequence, then from any position, there are 900 different places I can go. So I'm going to move around in this space. And question is, for this particular antigen, influenza HA, do my good antibodies fall on these preferred tracks, the high-speed trains, or to get to the high-affinity antibodies, do I have to take the high-speed train to a particular location and then take a local to get there? So what we're going to do is we're going to ask, does the system stay on these preferred mutational pathways? Is it sufficient to take only the mutable changes? And the answer is that it is. It's sufficient to take the most mutable pathways only. And this is a cumulative distribution of mutations as a function of the predicted mutability. Oh, you know what I'm doing? You have to see what this is, because this is something that only a PhD in physics would do. Okay, see this? If I press this, then I get my pointer. I was putting my finger over there because it's very comfortable. It's meant to do that. It's perfect. And I'm pressing and going, the battery must be dead. So, predicted mutability versus cumulative proportion. And this line in the center, this magenta line, tells you where the observed sequence would fall, the observed evolution throughout the entire tree would fall if there were no selection. And this tells you where it would, well, same thing over here. This is the accumulation of synonymous mutations, and this is the accumulation of non-synonymous mutations. So the point here is that the synonymous mutations are actually falling above this predicted line, and the non-synonymous are falling slightly below. So this is our first hint that we're staying on the superhighways or the high-speed lines, but it's only suggestive. The real answer comes from doing real modeling, and so this is where the real modeling comes in. And I won't belabor this, but this is the appropriate statistical model for mutations of type T with given gene G, position I, and so forth. And we measure the probability of having a mutation there by saying, what's the probability of getting a mutation there in the first place, and that's what all of this mess is. The subscripts are required because if you're moving in the tree, it depends what your ancestor is. So from node to node, you need a different index, unfortunately. But anyway, for the probability that any given nucleotide will be mutated is the product of the probability that it will be mutated to start with times the probability that it will be preserved in selection. And going through all of this, we can show clearly that the high affinity solutions that we have all fall right on the superhighways. That it would seem that this antibody is preset to find good antibodies for influenza. Now, there are lots of caveats there, and we're going to be studying the same kind of process for other antigens. But the main take-home lesson here is that affinity maturation works really well. And I think part of the reason it works really well is that one never has to explore the entire huge genotype space. We are restricted to exploring a very small subpart of that space, but by some remarkable evolutionary design, we are restricted to the space where the good solutions are going to be found anyway. Okay, now your attention may wane. Okay, because the rest of this is going to be relatively straightforward and won't be quite as abstruse as all of that. This is going to be much more direct. So we're interested in broadly neutralizing antibodies against HIV-1. And at the time that we wrote this paper 2 years ago, there were about 56 broadly neutralizing antibodies that had been isolated from patients. And we were interested in some of their properties. And as I mentioned before, if you look at the percentage of mutations in these antibodies, we find that the most mutated of them all, such as VRC01, are up around 36%. But there are plenty of others, 32%, 32%, 32%. And the very lowest of all is 9.3%. The average is around 20% or so. And if you look at the mutation frequency in a random collection of heavy chains, in this case it's 37,000 heavy chains that we downloaded from GenBank, the mutation frequency histogram looks like this. And this mark where you can't see any more bars is at 16%. See, so broadly neutralizing antibodies start at extremely high levels and then go way, way off to the end. Again, telling us that affinity maturation is a really key process. So what my colleagues at Duke did, spearheaded by Larry Liao and Barton Haynes at the Duke Human Vaccine Institute, they used the same techniques that I mentioned before, this sort of single cell isolation. Only they did it, in this case, on cells that had been sorted for capacity to bind the envelope protein on HIV. So they found 4 antibodies that were related to each other clonally. They called them CH104, 103, 106, 105, and they had nice broadly neutralizing properties. And I did the reconstruction, I found the unmutated common ancestor, but you notice how far away these antibodies are from this unmutated common ancestor, and that's because the mutation frequency of these guys is about 14%. It's pretty far away. And so I couldn't really be sure of what that unmutated common ancestor was. And so what we did was we made primers that would pick out antibodies with the appropriate V gene and J genes, and went in and did high throughput sequencing on the same patient from whom these broadly neutralizing antibodies were obtained. And that means that we were able to recover all of these other heavy chain genes, and fill out this phylogenetic history much more carefully. And of course, we had to identify which genes were really clonally related, and that's another story. But the point is that once we had all of this information, we were much more able to reconstruct the unmutated common ancestor appropriately. Now, this is color-coded by sampling time. So blood samples had been taken over a 2-year period in this individual, actually 3-year period. And so what we can see is that these broadly neutralizing antibodies had an ancestor. It was very close to the unmutated common ancestor, so it just had a few mutations in it, that already appeared at 14 weeks post-viral transmission. And this is really important because these guys were not isolated until more than 2 years after transmission. And it has been regarded as a given truth that broadly neutralizing antibodies don't arise until much later on. Well, and that's also true for this person, but their predecessors are there very, very early in the beginning. So we can now study this whole history in the same way that we studied the history of the influenza clone and learn a great deal from it. So let's just look at the binding affinity here. So again, we're looking at the binding affinity. The unmutated common ancestor has actually fairly good binding to the virus that was transmitted to this individual. So we were able to sample virus and antibody at various time points. And the original form of the antibody, which we inferred, binds to the original form of the virus. Good. And then as the antibody evolves, its binding strength becomes greater and greater and greater until its binding becomes quite tight. And furthermore, it's binding to a virus that it has never seen before also rises quite substantially. So that by the time we're out here with the observed antibodies, not only is it binding its own autologous virus very well, it's also binding an unrelated virus very well, so that it's acquiring both potency and breadth during the course of this evolution. And now the final part of this is to look at the role of another component of somatic mutation. Again, thinking about these HIV-1 broadly neutralizing antibodies, not only do they have very high levels of somatic mutation, nucleotide substitutions, but it turns out that they also have lots of insertions and deletions. And it's known that somatic mutation is capable of generating insertions and deletions, but usually they come at a very, very low frequency. So about 1% of all somatic mutations are insertions or deletions. And if you pull antibodies at random from a person or from a database, you get about 1% of antibodies also give you insertions or deletions. But among these broadly neutralizing antibodies, the frequency of insertions and deletions is more like 50%. And so they seem to be playing an important role here as well. And now our question is, what is it? What we're going to do is we're going to look at this class of antibodies as a whole, characterize them. Then we're going to be looking at high throughput sequencing of patients who've been followed over many years. And then we're going to look in detail at this particular broadly neutralizing antibody called CH30 through 34. There are 5 antibodies. And they have a rather remarkable 27 nucleotide insertion. So this is an example of the kinds of events that I'm that I'm telling you about. This is the observed sequence for one of the broadly neutralizing antibodies. This is alignment to its unmutated V gene. You can see, first of all, that there are lots of mutations, but second of all, there's this big deletion. So all of this was deleted. Here's an insertion, and here's another insertion. So these things just occur all over the place. So we wanted to ask two questions. What's the frequency? Why is the frequency so large? And there are a couple of different possible answers. One of them is that people who end up making broadly neutralizing antibodies have some host factor that predisposes them to both things. Predisposes them to making insertions and deletions, but also to making broadly neutralizing antibodies. And if that's the case, we as vaccinologists are kind of in trouble, because we can't give the host factor to people who don't have it in order for them to make broadly neutralizing antibodies. Other possibility is that HIV infection itself induces insertions and deletions. And the other possibility is that there's nothing at all unusual going on. And this is just a natural occurrence that happens when the immune system is stimulated over and over and over again for years and years at a time. And so we did a study using high throughput sequencing of 75 subjects who gave up a total of 261 blood samples. And we looked at 3.8 million sequencing reads. And the bottom line is, I mean, you're free to look at these lots if you like. But the bottom line is that we can predict the rate of insertions and deletions entirely by looking at the total frequency of somatic mutations. In other words, there's nothing special about HIV broadly neutralizing antibody producers. There's nothing special about HIV infected individuals with regard to insertions and deletions. If you can tell me what their mutation frequency is, I can tell you what the insertion and deletion rate is. So the HIV infected individuals have their immune systems, their B cell arm, driven so hard that they have higher mutation rates, higher mutation frequencies, and as a result of that, we get higher insertion and deletion rates. Pretty simple, and thank goodness as a vaccinologist, because that means that all we have to do is drive somatic mutation and the rest follows. But what exactly follows? Why do we have these insertions and deletions? Do they serve a purpose? And the answer is, I'm going to skip that. I'll skip that too. The answer is that they do seem to serve a purpose, in this particular case anyway. So I'm going to leave us with this final story. If we look at the phylogram of the antibodies here, these are the mature antibodies, and each of these, boxed in blue, are sequences that we recovered through high throughput sequencing and synthesized in order to measure their properties. Now you notice that this guy here, called 2GNM7S, is just off the main branch of lineage from the unmutated common ancestors to the mature antibodies, and is the last antibody before this insertion-deletion event. And here's what that insertion-deletion event looks like for this particular clone. There's the unmutated common ancestor, and here's one of the mature antibodies, and there's a 6-base deletion down here, and a 33-base insertion up here. And if you look carefully, you can see that this insertion is a copy of this region here. So it's a tandem duplication. 33 bases were duplicated and 6 bases removed. And it occurred, we found no antibodies, there are no genes that have one or not the other, so they probably occur very, very close to each other, if not in the same individual. So we look at this gene, which is the last gene before, and that gene, which is the first gene after. And we make hybrids of the various combinations of insertions and deletions and point mutations and so forth that represent the intermediates between these two. And we study their binding properties. And what we find, oh, this came out terrible, sorry about that. Let's focus our attention on the on rate. So this is the rate at which the antibody antigen bind together initially. And you want that to be high. So starting at the starting at this one, 2JSKAU, the on rate is shown here, and then the next one is the observed 2GMM7S, and then these two guys are the synthetic constructs that we made by just putting in the duplication. And by putting in the duplication, we've increased the on rate by a factor of 10. And so this one change has brought about a tenfold increase in the on rate, and has actually also improved the off rate. Conversely, if we take the first gene after the insertion deletion event and we take out the duplications, we lose much more than tenfold. So the duplication was made, and then additional mutations were incorporated that, so to speak, took advantage of the presence of that deletion. So taking away the deletion, taking away the duplication now makes the antibody much worse. And similarly here for the off rate and the overall affinity. Bottom line, this particular event, this large-scale insertion-deletion event, was a crucial element of the development of high affinity for this antibody, and I won't linger on this, but also for the development of neutralization capacity for a wide variety of different viruses. So we're now trying to understand more clearly what the role is for insertions and deletions and somatic mutation, and looking at a number of other systems as well, in addition to HIV, influenza, and anthrax. And I'm not going to talk about any of this. I just want to get through to the end so that I can acknowledge all the folks that worked on this over the last few years with us. At BU, Fang Fang is now a junior faculty member who was a postdoc of mine at Duke. Kate Sawatsky is a graduate student. Rekha Bhaskarabatla was a bioinformatician at Duke. A whole slew of people, but I'm going to call out Bart Haynes, Larry Liao, Munir Alam, and Garnet Kelso. In particular, at Stanford, Scott Boyd and Andrew Fire did the sequencing. Peter Harbour at Los Alamos National Labs did the rendering in beautiful violet and purple colors for that 1 filogram. And of course, we were sponsored by a number of wonderful organizations, and in particular, NIAID. And with that, I'd be happy to take any questions. Yes?
I always thought the on rate of an antibody for a target was less important than the off rate in terms of developing high affinity antibodies.
Well, so why did you think that?
Generally speaking, the slower the off rate is, the longer will be the residence time of the antibody to the target.
True, but it has to get there first. And so we've now looked at the on rates and off rates for a lot of antibodies, and we've seen both types of affinity maturation, those driven by increased on rates, as well as those increased by decreased off rates. So we are seeing both.
So we must have the 3D structure of the ID protein, right?
I'm sorry? Oh, yes, we do.
So if you model this 32-base type insertion, can you find out what really is adding that really makes it a much better scientific number?
Yes, and in fact, we've actually crystallized that the antibodies and compared it to the structure of the unmutated ancestor. And frankly, it's very difficult to say. It's hard to look at and say why it has improved. We don't have a co-crystal of these antibodies with GP120, which is what we really want to
But
have.
it's doing more than HIV. It's also become better than plus antibody other viruses too.
Oh, no, I'm sorry. The other virus was another strain of HIV. Yeah, I'm sorry if I didn't make that clear. Yeah.
Okay, thanks very much, Tom.